All right, in this explainer, we're diving deep into the genetics of cervical cancer and a really fascinating discovery that is changing how we think about the HPV virus. So we've known for a long time that the human papillomavirus, HPV, is the main culprit behind most cervical cancers. It does this by literally inserting its own DNA right into our genome. But for years, a really key part of this whole process has been a total puzzle. Is it just a random, unlucky event where the virus happens to land in a bad spot? Or is there something more deliberate going on here? Scientists have seen all sorts of conflicting evidence, which has really led to this crucial question.
So, to solve this puzzle, a groundbreaking study took a close look at the tumors of 106 cervical cancer patients, basically searching for clues in the virus's genetic footprints.
And what they found was pretty remarkable. It turns out, HPV doesn't just have one way of breaking into our DNA. It has two. And this discovery revealed two very different suspects in the story of how this virus drives cancer. The researchers gave these two suspects some pretty fitting names: silent integration and productive integration. Okay, so think of silent integration like a dormant computer virus, right? The malicious file has been downloaded. It's sitting somewhere in a random folder on your hard drive, but it's not actually running. It's not causing any active harm. It's just there. Productive integration, on the other hand, is the complete opposite. This is like that malicious code has been inserted directly into your computer's operating system. It is actively running, it's hijacking programs, and it's corrupting the entire machine from the inside. So the really crucial point here is the difference between the virus simply being present in our DNA versus the virus being active and actually doing something destructive. One is quiet and seems totally random, while the other is loud and looks suspiciously deliberate. Now, to figure this out, scientists needed to see where these integrations were actually happening in our DNA. They looked at the exact points where the viral genome literally broke into our own. And at first glance, just looking at the DNA evidence, the integration sites look totally random. I mean, on this map of our chromosomes, you can see the marks showing where the virus got in are just scattered all over the place. This really seems to support that bad luck theory. But then came the aha moment. Researchers looked deeper. They checked which viral genes were being actively read into RNA, and the whole picture changed. Take a look on the left. That's a productive site. You see those orange bars? That's RNA activity. The virus is awake. But then on the right, in a silent site, there's nothing. It's completely silent. And this led to the key finding. Those silent integrations, they really were random. But the productive ones, no way. They were much more likely to happen in specific, vulnerable parts of our genome. This strongly suggests that these productive integrations aren't just accidents. They actually give the cancer cell a survival advantage. They're being selected for as the tumor evolves. So, what is the actual impact of one of these productive integrations? I mean, what does this active, selected-for virus actually do to the cell? Well, Exhibit A, just look at this chart. Tumors with productive integration show a huge increase in the activity of nearby human genes. The virus isn't just sitting there, it's hijacking the cell's machinery and forcing it to work overtime. This hijacking creates these bizarre, abnormal fusion genes, part virus, part human. You could call them Frankenstein genes, and they lead to the overproduction of HPV's most dangerous weapons, its oncoproteins, which are the proteins that cause cancer. And this slide right here, this is the smoking gun. Exhibit B. The data shows that tumors with productive integration have far more of those cancer-causing oncoproteins, specifically E6 and E7. These are the proteins that shut down our body's natural defenses and just drive the cancer's growth. To put it simply, more productive integration means more fuel for the fire. So what do all these molecular clues actually mean for real patients? This is the crucial link. When the researchers compared their findings with patient data, the trend was undeniable. As cervical cancer gets more advanced, moving from stage 1 to stage 3, the percentage of tumors with this aggressive, productive integration goes way up. The takeaway here is crystal clear. Productive HPV integration is not just a random event. It's a specific biological mechanism that gets actively selected for, and it is clearly associated with the tumor progressing and a worse prognosis. As the researchers themselves put it, making this distinction between silent and productive integration fundamentally changes how we view HPV's role in causing cancer. It's not just about whether the virus is present, it's about what it's doing. And this leaves us with a provocative final thought. If productive integration is a clear hallmark of more aggressive disease, could we develop new tests to look for it? This could potentially help us identify high-risk patients much, much earlier, and maybe even lead to more personalized, effective treatments. The story of HPV and cancer just got a whole lot more interesting.